Hello, everyone. So let's dig into our second week exploration into the biological psychology of religion. And what I've chosen for the bulk of our readings is Bruce Lipton's The Biology of Belief. And so let's dig into chapter 1 now. Lipton is a cellular biologist. He's a professor of cellular biology. And his theory, his ideas of how our human organism functions at the biological level, at the cellular level, is mirrored or replicated from the microcosm, micro meaning the smaller, to the macrocosm, the larger. So, for example, in theoretical physics, there's a concept called fractals in chaos theory. And fractals basically, a simple example would be if you take a head of broccoli and you look at the structure of the broccoli at the physical level, the naked eye level. And then you take a microscope and you look at this at a zoomed in level, you'll see smaller structures that resemble heads of broccoli. And we go from the larger level to the micro, from the macro to the micro. And what we see is a repetition of structure. You'll read in Chapter 3 of Lipton's text some ideas about crystals, salt crystallization in biology and chemistry. And this is very similar with the structural growth of crystals. If you look at crystals, there's a fractal growth, a replication in the macrocosmos of the microcosmos. The big world and the little, macro and microcosmos. So this is really something to consider about Lipton's work. He's looking at the cellular level of living beings, human beings, and he's looking at the behavior of cells within a given environment. He is then extrapolating that microcosmos to the macrocosmos and understanding this relationship between the organism on a big scale, on a macro scale, as related to the organism on a micro scale. And he's showing us that there's many behavioral and, if we want to say, character traits or characteristics at the cellular level that there is at the human organ level, at the macro level. Now, hopefully that'll help you to understand what's going on here. His writing is very accessible. He's telling us the story of how he is a professor of in the medical school at the University of Wisconsin, and fate had taken him to spend some time in the Caribbean, teaching at a number of medical schools in the Caribbean as well. Sounds like a nice opportunity to spend some time in tropical islands and make a living. Well, he was saying how this teaching experience, he's telling us the story of how his teaching experience in the Caribbean will come to be an analogy for the story that he's telling us about the cellular behavior. So please, I know, because reading this now for the third time, so there was the first time I read it a few years ago. The second time I read it, I got more out of it. And now I'm reading this for the third time with you all. Just like you are preparing your assignments each week, I'm preparing my assignments for you. And that involves me having to present these readings in an intelligent way and a comprehensible way, an accessible way. Fortunately for us, Lipton is making it really accessible. And admittedly, he gets into some pretty deep biological concepts here. He's not only asking us to understand biology as we learned it in high school or maybe in a class in college, but he's also asking us to reconsider biology in a new way. For example, understanding the differences between Darwinian evolutionary theory and Lamarckian evolutionary theory. We're going to go more into depth in this because this is an important aspect of the history of psychology and evolutionary psychology, which we're going to be spending more time with later in the course. And understanding how one approaches the understanding of biology will ultimately pre-consider, predictate, pre-establish how the phenomenon looks. So in other words, if we approach understanding biology through a Darwinian evolutionary perspective, we get a certain outcome, a certain framing of the picture. How do I do that? There it is. There's the frame. And if we take a Lamarckian perspective, we get a different look at things. There's one aspect and this is something to think about, and you'll read about these differences. And I think he illustrates it very clearly, the differences between the two. Related to that shift from a Darwinian to a Lamarckian way of thinking. And actually, I should illustrate, this isn't necessarily a changing and an abandoning of the Darwinian approach to evolutionary cellular biology, but it's a synthesis. It's a combining of the two. We have the Darwin, we have the Lamarck, and what he's presenting us is this Darwin/Lamarck view. That following from that is the shift of the center of the human living beyond humans, all species, but specifically in our discussion, the human communication method of genetics, passing on. Genes are a communication system. We have three communication systems in biological psychology. We have the nervous system, which is in essence communicating from sensory organs to the brain, and from the brain to the muscles. We have, for action, we have the endocrine system, which is communicating from glands through hormones, through the bloodstream, to various systems of the body. And we have the genetic communication system, which is transmitting information from generation to generation. So the genes are centered in this area of the cell called the nucleus. And one of the things that he describes in his writings is the shift from thinking of the cell nucleus as the brain to thinking of the cell nucleus instead, as he says, the gonads, as the reproductive methods of the cell. He shifts the intelligence, the brain of the cell, to the cell wall. Some of you are reading this and you are saying, I have no interest in this whatsoever. Others are finding this absolutely fascinating. So I think it's important, as I keep in mind discussing this with you, that as we talk about the biological aspects of psychology, of the human condition, that how it's received, that just this approach, how it's received is highly dependent on the person who's receiving it. It is an aspect of psychology. It's becoming even more important in the 21st century. When I was in graduate school, we had we referred to this as biological psychology, and we had courses that were mostly on neuroanatomy, areas of the brain, and we got into also functioning at the cellular level, so neurons, nerve cells, Hebb learning theory, all these ideas, how neurons fire, serotonin neurotransmitters, etc. In the past few years, courses have opened up into subatomic psychology. So looking at how the biology within the cell, looking at not only at a cellular level, but more magnified into the subatomic level, looking at protons, neutrons, electrons, etc., how these atomic, subatomic aspects at the micro level translate into the macro level. This is what's happening here. And so, as I said, some of you dig it and some of you don't, and this course is to expose you to a multiplicity of different approaches to understanding human phenomenon. So that's where we're at in the biological psychology of religion. As we have already looked at humanism, we'll look at psychoanalysis and others. So, that being said, he's asking us to shift our way of thinking from the nucleus being the brain to the nucleus being the gonads of the cell. What do the gonads do? It actually makes very good sense here. The gonads replicate. They pass on. They make more, just like our gonads make more. They make children. Well, these gonads dictate the making of other cells. That's the cell nucleus. That is what he is really concentrating on. Well, he presents the big picture in chapter 1. In chapter 2, he's really concentrating on this shift regarding the nucleus and the cell. This shift from viewing the nucleus as the brain to the nucleus as the gonads. In chapter 1, he presents this idea. In chapter 2, he goes into detail to explain the importance of the environment. And to give you an example of this, taking the understanding of the nucleus as the brain would be akin to our thinking of genetics as the thing that dictates us. So it's very popular today, and it's a little bit of a holdover from 19th century thinking that somehow we are determined by our genes. It's very popular to talk about a genetic cause, hard determinism, as we've discussed. We know from our previous reading that this is not a responsible position to be taking, to say, I act this way because my genes dictate it. We know instead that we understand what James called self-determinism, that there might be an influence, but ultimately there is a cognitive aspect. This is what cognitive psychology is all about. A cognitive aspect to make decisions on how to deal with certain desires, impulses. And ultimately, where there could be a biological pressure, a biological drive or desire as we'd experience, ultimately our decisions are what dictate our behaviors. Those decisions are based on our perceptions. I've given you the gist of his entire premise here. Our decisions are based on our perceptions. If we hold certain beliefs and certain attitudes about desires, biological pressures, genetic predispositions, we then we understand those desires, those predispositions, those pressures in uniquely different ways based on our beliefs. And then we act accordingly to those beliefs. And if we're acting within accordance to those beliefs, we have a certain emotional response. And if we act In opposition to those beliefs and attitudes, we have another emotional response. We call it guilt, cognitive dissonance, a plethora of different framings of how we understand that. So when he is discussing this idea of a cell wall, the cell membrane, he likes to play with this word mem/brain, as the intelligence of the cell, what he is saying is, well, what is our membrane? What's the membrane that we have at the human level rather than the biological, at the cellular level? What is our membrane? It's the skin. It's the skin. And what do we have coming through the skin? We have the skin, the sense of touch and pressure. We have the sense of temperature, warm and cold. Combine the two, we have hot. In addition, we have other membranes. The tongue is a membrane, the surface of the tongue. And on that membrane, we have receiving cells, receptor cells. We call them sensory cells, taste buds. We have the eyes, another membrane. The retinal tissue is a membrane. We have the ears, not the ears here, but the inner ear. Maybe more specifically, the eardrum is a membrane. And of course, we have the nose for smelling. Olfactory bulbs are a membrane. If we understand that as we go into the world and we interact with our environment, our body, our flesh envelope, our sensory organs are coming into contact with stimuli in the environment. The conditioning of our sensory apparatus will largely dictate, at one level, how we perceive, sense and perceive the environment. Let me give you an example of this. If you happen to be someone who is an artist, visual artist, or an interior decorator, or a painter, something like this, a fashion designer, someone who's dealing with colors, you will have daily exposures, hours of exposures to different colors, not just green, but different shades of green that have different names assigned to them. Or if you're into computer graphics, different hex codes assigned to different colors. Hours turn into days, days turn into weeks, weeks turn into months, and months turn into years. And in a lifetime, your eye will become educated to colors. You will be able to see shades and differences in colors that someone who doesn't spend time with the colors will not see. You'll see two shades of green, you'll say, which do you like? And your friend will say, they're exactly the same. This is because you've educated your senses. You've educated your sense of sight. If you're a musician, and on this one I'll speak first-hand experience. If you're a musician, you spend hours a day, days a week, weeks, a month, months of years, cultivating, educating the ear. If I go to a certain event or something, I hear someone sing, my ear is so finely tuned that I can hear inaccuracies in pitch, out of tune, in other words, someone singing out of tune. And often a friend will say, isn't that an amazing singer? I've heard amazing singers many times. I've heard intonation, and I will hear problems in the intonation the other person just can't hear, simply because I've educated my ear. Those of you who've grown up in different cultures, let's take, for example, Indian or Chinese music. Your ear, now if you grew up in that culture from birth through your first 6 years of life, your ear has been exposed to musical scales that someone who grew up in a Western culture is likely has not been exposed to. If you come from an Indian culture, a Chinese culture, you know exactly what I'm talking about. There are pitches that exist in Indian music and pitches that exist in Chinese music. There are pitches that exist in African and West African music that do not exist in the Western concept of diatonic music. Do, re, mi, fa, sol, la, si, do, si, la, sol, fa, mi, re, do. And in between those, in Western music, we categorize those in half steps. Do, re, mi, fa, sol. That's all the keys on the piano if you play them. In Chinese music and Indian music, there are tones in between those black and white notes on the piano in Western music. And whereas we would hear those tones as in tune or out of tune relative to our C, C sharp, D, E flat, E, F, F sharp, etc., in Indian and classical music, they are distinctly discernible tones in their own right that aren't seen as either in or out of tune. They're seen as distinctly different tune. As Westerners, we hear the music and it sounds maybe runny. It sounds maybe at moments out of tune to the untrained Western ear. But if you grew up with this music, or if you expose yourself to this music repeatedly, you begin to learn to educate the ear, to learn the subtle differences of these pitches, and it changes what you hear. Instead of hearing something as out of tune, you hear something as perfectly in tune and intentional, decorative of other tones, as happens in the raga, in the sitar playing. Okay, what is this illustration about? The illustration is this. Our membrane, the cell wall, the sensory apparatus of our skin, our eyes, our nose, our tongue, and our ear are our membrane to the environment. And that membrane is intelligent. It changes. And it changes in relationship to the environment. The more non-Western music you're exposed to, the different and shift in understanding of that music becomes. In this way, someone who is at the cellular level, there's a certain neurotransmitter present in the system, and when that neurotransmitter is flooded into the system, the cell wall adapts by creating more receptor gates. It's an intelligent form. When the neurotransmitter is not present, those receptor gates are pruned. They are purged from the cell wall. They're not used. We know from neural pruning, the word I just used, pruning is taken from neural pruning, that in the first 6 months of life, what is not used in neural circuitry is pruned away, not used. We lose neurons. And what is used becomes reinforced. I hope this is all helping to bring to you this understanding of Chapter 2. It's the environment stupid, he calls it. What he's illustrating to us in a very technical way is the adaptation of the membrane to the environment at a cellular level. And in Chapter 2, he's going into detail to show this parallel with our social level, the human as a membrane. Now, this idea of seeing human characteristics in non-human entities is called anthropomorphism, and as he points out, this is a taboo in science. And this is an illustration to show us that if we shift our belief about how we understand something, our understanding of something shifts. So if we momentarily say, okay, I know cells aren't humans, but if I take this analogy as one that will open up new possibilities in thinking for me, what will those possibilities become? The only way we can discover that is if we take the leap and give it a try. So this is hopefully helping you to make sense of these chapters. Now, what I'm going to ask you to do is not to get overwhelmed by the technical aspects of the cellular biologist. The guy is really going into depth, and he's doing his best to give you great illustrations. One of my favorites is in chapter 3. He gives all these wonderful illustrations in chapter 2. But in chapter 3 in particular, he gives the example of how let me get to this, how the membrane works. He told me, here's a quote, he told me that when the cultured cells you are studying are ailing, you look first to the cell's environment, not to the cell itself. My goodness, can we understand that as an analogy or an allegory for therapy itself? When a young child comes into the office brought by their parents, repeatedly what I've seen is the child is a symptom of what's happening at home. The parents saying, what's wrong with my child, while they acting this way and pointing the finger at the child, and we sit down and the first thing we do is family therapy. Because we see this as a unit, not as the child as the problem, but as the child as a reflection of the environment at home. This is exactly what's being described here. He goes on to say, cell biologists gained insight into the amazing abilities of the cell membrane by studying the most primitive organisms on this planet, these prokaryotes. And yes, I did look that word up to know how to pronounce it properly. And so he takes these little cells and he explains to you through these cells what he means by intelligent membrane. I did my best to explain to you as psychologists what that means. So here's the illustration I want to give you about the cell membrane. Bread, butter, olives, and pimentos. He says, I came up with a gastronomic treat to illustrate the basic structure of the membrane. The treat consists of bread and butter sandwich. To further refine the analogy, I added olives. Actually, my instructive sandwich features 2 kinds of olives, some stuffed with pimentos and others pimento-free. You know, pimento is that little red pepper in the olive. Gourmands don't groan. When I've left this sandwich out for my lectures, repeat members of the audience have asked me where it went. Here's an easy experiment to show you how the sandwich membrane works. Now he's using this, and he gives pictures in the book, and it's a really remarkable way to understand what he means by intelligent membranes. Make a bread and butter sandwich, the moment olives free. This sandwich represents a section of the cell membrane. Now pour a teaspoon, so this is the membrane of the cell. Bread, butter, bread. Now pour a teaspoon of colored dye on the top of the sandwich, and what happens is, illustrated below, the dye seeps through the bread but stops when it gets to the butter because the oily substance in the middle of the sandwich provides an effective barrier. Now let's make a bread and butter sandwich stuffed with unstuffed olives. These are olives without pimentos and no pits inside. They're pitted, so they're hollow inside. Now when we add the dye to the bread and slice the sandwich, we see a different result. When the dye hits the pimento stuffed olive, it stops as surely as it stopped when it hit the butter. Apparently he changed his mind and he's putting pimentos in some of these now. But when the dye reaches an olive without the pimento, the pitted olive provides a channel through which the dye can flow freely across the middle of the sandwich and through the bread to the plate. The plate in this analogy represents the cell's cytoplasm. By bypassing through the pimento-free olive, the dye penetrates the buttery layer to reach the other side of the membrane of the sandwich. The dye has successfully navigated the formidable fatty membrane barrier. It is important for the cell to allow molecules to break through the barrier because, in my sandwich analogy, dye represents life-sustaining food. If the membrane were simply a bread and butter sandwich, it would provide a fortress-like barrier that keeps out all the cacophony of innumerable molecular and radiant energy signals that make up a cell's environment. But the cell would die if the membrane were such a fortress because it would get no nutrients. When you add the pimento-free olives, which allow information and food into the cell, the membrane becomes a vital, ingenious mechanism, enabling selected nutrients to penetrate the interior of the cell just as a teaspoon of dye made its way to the plate. In real-life cellular biology, the bread and butter portion of the sandwich represents the membrane's phospholipids, one of the two major chemical components of the membrane. The other major chemical components are the olive proteins, which we'll get to below. I call phospholipids schizophrenic because they are composed of both polar and nonpolar molecules. Okay, so I'm going to stop there. What he's illustrating to us is very similar in Introduction to Psychology, the concept of receptor gates in the neural cell either increasing or decreasing based on the availability in the environment of a certain specific neurotransmitter. If you remember this, when there's more neurotransmitter, there's more receptor gates. When there's lesser neurotransmitter available in the environment, there's fewer neurotransmitter gates. On a social level, let's say you're taking all these college classes. The more college classes you get, the more adept you become at processing information and managing how to get that information into your mind and fulfilling the assignments and tests and multiple all that stuff. And you get in shape, in other words. You go out and you want to do something physical. The first few days you're trying it are very difficult. A few weeks later, after daily practice, you're more adapted to it. This is what's happening with cellular walls. It's what's happening, as he's illustrating, with it's an intelligent membrane. It adapts to the environment. So let's move to the integral membrane proteins. The olives in our sandwich are a truly ingenious part of the membrane. These proteins allow nutrients, waste material, as well as other forms of information to be transported across the cell membrane. The protein olives allow not just any old molecules to get into the cell body, only those molecules necessary for the smooth functioning of the cytoplasm. In my sandwich, the olives represent integral membrane proteins. He's calling these IMPs. Integral membrane proteins. These proteins embed themselves into the butter layer of our membrane, just as I have embedded olives in the illustration. He goes on to tell us how the brain works. He's going to show us how this applies at the brain level. Once I understood how IMPs worked, I had to conclude that the cell's operations are primarily molded by its interaction with the environment, not by its genetic code. So this is what I explained in the first part of this video. There is no doubt that the DNA blueprints stored in the nucleus are remarkable molecules which have been accumulated over 3 billion years of evolution. But as remarkable as these DNA blueprints are, they do not control the operations of the cell. Logically, genes cannot pre-program a cell's organism's life because cell survival depends on the ability to dynamically adjust to its ever-changing environment. So what we're illustrating here is something really that evolutionary psychologists do know, that we have certain influences over thousands of years of biological adaptation that are influences on our life, but we also have an evolutionary process that is more, as we would say, more oriented to the present moment. Faster might be a word. In other words, you are adapting every day to circumstances in your environment that in that adaptation, that managing of your environment on a daily basis, on an hourly basis, takes into consideration pressures coming from thousands of years of genetic predispositions, genetic pressures, and consciousness, belief, and how you manage an individual moment has more to do with your beliefs about the past and the present and the future, as we'll see. Let's put the membrane to the same brain test to which we put the nucleus. When you destroy the membrane, the cell dies, just as you would if your brain were removed. Even if you leave the membrane intact, destroying its only its receptor proteins, which can be easily done by digestive enzymes in the lab, the cell becomes brain dead. That's like turning off your sensory organs. It is comatose because it no longer receives environmental signals necessary for the operation of the cell. The cell also becomes comatose when the membrane's receptor proteins are left intact and its effector proteins are immobilized. So I'm going to skip ahead to get to some punchlines. I skip ahead for our purposes because I can't read the entire chapter to you. The secrets of life. As you've learned in this chapter, scientists have recently made great progress towards the unraveling and complexity of the simple-looking membrane. But even 25 years ago, the rough outlines of the membrane's functions were known. In fact, it was in 1985 when I first realized how understanding the workings of the membrane could be life-changing. My eureka moment resembled the dynamics of a supersaturated solutions in chemistry. The solutions, which look like plain water, are fully saturated with dissolved substances. They are so saturated that adding just one more drop of substance causes a dramatic reaction into which all of the dissolved materials instantly coalesce into a giant crystal. He goes into detail about his personal eureka moment of this. In 1985, I was living in a rented house in the spice-drenched Caribbean island of Grenada, teaching at yet another offshore medical school. It was 2 a.m. and I was up revisiting years of notes of the biology, chemistry, and physics of cell membrane. At that time, I was reviewing the mechanics of the membrane, trying to grasp how it worked as an information processing system. This is when I experienced a moment of insight that transformed me not into a crystal, but into a membrane-centered biologist who no longer had any excuse for messing up his life. At that early morning, I was redefining my understanding of the structural organization of the membrane, starting first with the lollipop-like phospholipid molecules and noting that they are arranged in the membrane like a regimented soldiers on the parade in a perfect alignment. By definition, a structure whose molecules are arranged in regular, repeated patterns is a crystal. That's the fractals I was mentioning. The crystals that most people are familiar with are hard and resilient minerals like diamonds, rubies, and even salt. Excuse me. The second kind of crystal has a more fluid structure, even though its molecules maintain an organized pattern. Familiar examples of liquid crystals include digital watch faces and laptop computer screens. To better understand the nature of liquid crystals, let's go back to those soldiers in the parade. When the marching soldiers turn a corner, they maintain their regimented structure, even though they're moving individually. They're behaving like a flowing liquid, yet they do not lose their crystalline organization. The phospholipid molecules of the membrane behave in a similar fashion. Their fluid crystalline organization shows the membrane to dynamically alter its shape while maintaining its integrity, a necessary property of a supple membrane barrier. So in defining this character of the membrane, I wrote, the membrane is a liquid crystal. Then I started thinking about the fact that a membrane with just phospholipids would be simply a bread and butter sandwich without the olives. In the experiment described earlier, the colored dye could not get through the lipid butter layer. That bread and butter sandwich is a non-conductor. However, when you include the IMP olives, you realize that the membrane conducts some things across while leaving other things out. So I continued writing in my description of the membrane by adding the membrane is a semiconductor. Lastly, I wanted to include my description the two most common kinds of IMPs. These are the receptors and a class of effectors called channels because they provide an all-important means for the cell to let nutrients let out waste matter. I was about to write that membrane contains receptors and channels when I realized the synonym for receptor is the word gate, which you all are familiar with from Introduction to Psychology class. So instead, I completed my description by writing, "The membrane contains gates and channels." I sat back to review my description of the membrane. The membrane is a liquid crystal semiconductor with gates and channels. What hit me right away was the fact that I had recently heard or read the very same phrase, though at the moment I didn't know where I'd come across it. One thing was for sure: I was not in the context of biological science. It's interesting. Some psychologists in different perspectives would say we're not in the realm of psychology. The biological psychologist would disagree. As I leaned back in my chair, my attention was drawn to the corner of my desk where my new smiley-face Macintosh, my first computer, was parked. Lying beside the computer was a copy of a bright red book called Understanding Your Microprocessor. I had just bought this non-technical paperback guide to how computers work from a Radio Shack outlet. I grabbed the book and found in the introduction a definition of a computer chip that read, "A chip is a crystal semiconductor with gates and channels." For the first second or two, I was struck by the fact that the chip and cell membrane shared the same technical definition. I spent several more intense seconds comparing and contrasting biomembranes with silicon semiconductors. I was momentarily stunned when I realized the identical nature of the two definitions was not a coincidence. The cell membrane was indeed a structural and functional equivalent of a silicon chip. So here's what we take from this. He's not saying, as in cognitive psychology and information processing theory, he's not saying that the brain is a computer. He's not saying that the personality structure or the behavior or the way we think is a program that runs on a computer, like happens in cognitive information processing theory. He's saying there are similarities between the two, and looking at one, we can gain insights into the other. That's the method of theory. It's not saying that the brain is the hardware. It's saying the brain is like hardware. He goes on, "I had been trained as a nucleus-centered biologist, as surely as Copernicus had been trained as an Earth-centered astronomer. So it was with a jolt that I realized that the gene-containing nucleus does not program the cell. The environmental data is entered into the cell via the membrane's receptors, which represent the cell's keyboard. Receptors trigger the membrane's effector proteins, which act as the cell's computer or central processing unit." He concludes by saying, "I'll get to the spiritual part of the story in the epilogue." So he's not giving us the whole punchline of this book yet. "For the moment, let me reiterate the lessons of the magical membrane, which put the control of our lives not into the genetic roll of the dice at conception, but into our own hands. We are the drivers of our own biology. Just as I am the driver of this word processing program, we have the ability to edit the data we enter into our computers, our biocomputers, just as surely as I can choose the words I type. When we understand how IMPs control biology, we become masters of our own fate, not victims of our genes." So I want to conclude this by just saying I've selected some central ideas to help you to understand. I'm going to ask you not to get bogged down in the technicalities of his explanations. Read them, know about them, be aware of them, but that's not the gist of what's going on here. This is just an introduction to biology of psychology, a biology of religion, psychological biology of religion. So you have to know what is useful at this moment and what might be useful later in life if you're interested in this. At this moment, expose yourself to the first time. Expose yourself to the general concepts, like I did the first time reading. If you're interested, you go back and you read it again. And you research the little aspects that he's discussing and proposing. So at this point, we are just looking at big picture stuff. So that is that's the introduction, first 3 chapters of Bruce Lipton's The Biology of Belief, and now things are about to get very interesting in the next portion of the book.